we have already seen that variation is an extremely important component in evolution, and without variation evolution cannot really proceed. However, there is one more thing about variation that we have to appreciate, which is that it is not sufficient to have a lot of variation. The organism or the species must have ways of generating new variation. Why is that so? That is because think about the conditions in which an organism or a population finds itself in nature. Is nature constant? No, it is always changing. You have new pathogens arising, you have new predators arising, you have the temperature changing, you have rainfall changing, you have the availability of food changing, etc., etc. Therefore, over time an organism or rather a population is continuously facing new challenges, and in order to be able to meet these challenges, the population has to have ways to generate new variations. So, now that is what we are going to discuss next. What are the ways in which new variations are generated by populations? Now, there are four major ways in which new variations are generated. These are recombination, horizontal gene transfer, also called lateral gene transfer, introgression and mutations. Now, these are all topics that you have seen or learned before in a basic genetics course. So, what I will do is I will kind of go through them a little fast. I will not talk about the mechanics of any of the topics, but I am in general going to talk about how these things, these modes of generating new variation, how they relate to evolution. So, first, recombination. So, what is recombination? As we know, recombination is the exchange of parts of chromosome during the process of meiosis 1, also known as crossing over. So, this is showing you the process of recombination. Now, what recombination does is that it reshuffles the alleles from the parents in various combinations. Now, we already know that various genes at various loci, they are interacting with each other in terms of their function, which essentially means that the traits of an organism are dependent on precisely which alleles are there in what combination. And what recombination does is, it is continuously, through this reshuffle, it is continuously creating new genetic combinations, which results into new phenotypes, and therefore the total diversity of traits in the population goes up tremendously. Now, it is estimated that in any sexually reproducing organism, this kind of variation that is arising due to recombination is a huge portion of the total variation available. Now, one thing one has to keep in mind is that recombination is actually a double-edged sword. What do I mean by that? Remember, recombination can bring alleles in combination with each other, but at the same time it can also take away or break favorable combinations. So, in that sense it is a double-edged sword. It is continuously creating and breaking these favorable allele combinations. The second very important thing is what is known as horizontal gene transfer or lateral gene transfer. So, as we know that in most organisms, most of the time the genes that we have, they are coming from our parents, right? But there are certain situations under which you can have transfer of genes without reproduction across organisms, and in many cases this transfer of genes can even happen across species boundaries. So, these are what are known as horizontal gene transfers. Now, one thing we have realized is that horizontal gene transfer is very, very common in prokaryotes. However, there are enormous number of examples also known from the eukaryotic world. For example, much of the drug resistance that we find in microbes today, you know that it is a huge problem. You go to any hospital, most of the microbes that come up over there, many of them are actually multi-drug resistant, and it is thought that we are going to run out of antibiotics to give to our patients very soon, until and unless something major can be done. Now, this process, a lot of it is actually happening due to horizontal gene transfer. So, basically you treat one pathogen with your latest antibiotic, but that pathogen ends up getting resistance to it, developing resistance to it, and then that gene from that pathogen goes into other pathogens for which you have never used that antibiotic, but they end up getting that gene anyway, and therefore on their own they end up becoming multi-drug resistant. The same thing happens for pesticide resistance as well, particularly in bacteria. So, what is the implication of horizontal gene transfer for evolution? Sorry, before we go there, there is another form of horizontal gene transfer which is known as introgression. So, in horizontal gene transfer, what happens? A few genes are transferred, across individuals or across species boundaries. Now, in introgression, you have two different species mating and there is a formation of a hybrid. And this hybrid, typically we know that the biological species concept says that if you have two species, they should not be able to mate with each other. Now, obviously, that is not the species concept we are using here. We are primarily talking about the morphological species concept. Now, most of the time, the hybrids are infertile or they have very low survivorship or they are able to produce very few babies. But sometimes you end up having hybrids which are fertile, and when that happens, and if that fertile hybrid is now able to breed with one of the existing partners, and then it is able to pass on the genes from the donor species into the recipient species. And once this happens, this can keep on happening generation after generation, and at some point the genes from the donor species will end up getting incorporated into the gene pool of the species of the recipient species. In this process, obviously you can have more than one genes spreading into the recipient species. So, for example, a very famous example, if you look at the modern humans, about 1 to 3% of the genes that make up our genome, depending on which population you are talking about 1 to 3% of that genome is actually coming from Neanderthals. Okay. So, basically the assumption is that at some point Homo sapiens mated with Neanderthals and then their genes now have propagated into the human gene pool, and even after so many thousands of years it is still identifiable. Now, what are the evolutionary implications of horizontal gene transfer or introgression? So, the evolutionary implication is very interesting. So, normally you remember when we were drawing phylogenetic trees, we got trees like this. These are what are known as branching trees, where each node has only one ancestor. So, the whole idea is one species is giving rise to another species and another species and so on and so forth. So, in this kind of a scenario, you get a tree which looks like this, which looks resembles the branches of a tree, as you can see over here. However, when you have horizontal gene transfer, what this is implying is that in the context of where your genes are coming from, you can have more than one ancestors. So, some of your genes are, let us say, coming from Homo sapiens, but some of your other genes are coming from Homo neanderthalensis. So, if you have a situation like this, then you end up having a tree which is known as a reticulate tree. So, here for example, you can see not only do you have this kind of bifurcating branches, but you also have connections between the branches. For example, the ones shown in orange and green, and these other ones which are shown in thin blue branches going horizontally. So, all this is showing is that the ancestry of one species can be from multiple species. Now, at some point, some people were very intellectually bothered by it, and almost to the point of claiming that oh my God, Darwin's theory is wrong. However, I do not think that is entirely correct, because even though you might end up having a few genes which is coming from a different ancestor, the majority of your genes are still coming from one ancestor. Therefore, that is one thing which you can see in this particular branching tree, that the thickness of the branches are very different, right. So, the horizontal branches, they are much thinner, showing that the contribution along that axis is very less, whereas the vertical branches are much thicker, saying that much of your genome is actually coming from that route, so through what is known as vertical transmission. Okay. So, because of this, in terms of the process of descent with common modification, horizontal gene transfer does not play any role over there. All it ends up showing is that the way you interpret your trees needs to be probably slightly different, and you have to acknowledge that some of the genes that an organism might have might come from a completely different species compared to the one for which the vertical tree is being drawn. So, that is all that is happening there, all that is there to it. And as I said, in a nutshell, it implies that phylogenetic trees sometimes can be reticulate instead of branching, and in no way does it pose a challenge to Darwinian descent with modification. Okay. So, these are the three relatively less important ones done, only relatively, but the one that has been most studied and the one that contributes maximally to the genetic variation is mutation. So, what we will do is a quick revision of various kinds of mutations, again without going into the mechanics of it, and once we are done, in the next discussion we are going to look at the certain properties of these mutations that make them very interesting from an evolutionary biology point of view. So, prior to that, quick revision of mutations. So, mutations can happen at various levels. At the level of individual bases, you can have what are known as point mutations. You can have mutations at the level of the entire chromosome, or you can have mutations at the level of the entire genome itself. So, we will study these one by one. At the level of individual bases, you can have a change, an alteration at the level of a single base, or you can have an insertion, or you can have a deletion. A new base can be inserted, or a new base can, or a base, existing base can be moved out. So, functionally speaking, in terms of their effects, the mutations can be divided into three kinds of, three types. One is what is known as silent. So, when the change in the ATGC sequence is not changing the amino acid, that is when you get a silent mutation. Why does that happen? Because we have redundancy in the genetic code, because in many cases the same amino acid is coded for by multiple codons, and therefore, if your mutation is such that you have gotten into a synonymous codon, obviously the amino acid is not going to change, and that is why it is going to be a silent mutation. Similarly, you can have a missense mutation, where due to a mutation, due to a insertion or change, different amino acid is coded for. And finally, you can have what is known as a nonsense mutation. You know that there are three codons which are stop codons, and if a mutation is such that a stop codon is generated, then that is where your translation is going to be truncated, and therefore, that is you will get a smaller length of a polypeptide chain. I am simplifying this. Actually, there are many other kinds of nonsense mutations that are possible, but this is for a basic revision, this should be enough. Then at the level of individual bases, you can also look at what exactly is changed. So, when the number of bases remain the same, which basically means simply a change has happened, in that case there are two possibilities. One is what is known as transition, where you have a purine to purine or a pyrimidine to pyrimidine change. So, adenine becoming guanine or guanine becoming adenine, that is one. Similarly, C to T or T to C, all these are transitions. When you have a purine to pyrimidine or a pyrimidine to purine kind of a change, that is what you call a transversion. So, in this particular diagram, A, C, T and G, these are the 4 nucleotides. So, if you go along the diagonals, which is A to G or G to A or C to T or T to C, then that is what is called a transition. Anything else along the 4 sides of the square, it is going to be a transversion. So, just if you remember this diagram and remember that alpha is along the diagonals and beta is along the sides, then alpha equal to transitions, beta equal to transversions. This diagram is a simple way of keeping this in mind. What happens when the number of bases are altered? How does this happen? This will happen if you have an insertion or a deletion, in del as we call them. And when an insertion or deletion happens, sometimes but not always, you have what is known as a frame shift mutation. What is a frame shift mutation? So, you know that our codons are in triplets, right. So, 3 nucleotide bases together form a codon. So, if you have deletions or insertions which are not in multiples of 3, then the reading frame from your polypeptide chain is going to be altered, and when you have that, then there can be a huge change in terms of what is translated at the end. So, this is what is known as a frame shift mutation, and you know, a frame shift mutation is rather well critical. Many a times, for example, certain kinds of cancers etc. are caused by these kind of mutations. So, from this we move to mutation at the level of chromosomes. There are 7 major types, we are showing you all over here. So, you can have what is known as a deletion. So, in deletion you can see that a particular fragment of the chromosome simply gets deleted, or you can have a duplication where a particular fragment of the chromosome is duplicated and then it gets added right at the same place, as you can see in the second figure over here. Or you can have an inversion where a particular fragment of the chromosome is cut out and then it gets inverted and then it gets inserted at the same place in the chromosome, as you can see here in the middle figure. Or you can have a fission where you have a chromosome and which divides into 2 parts and becomes 2 separate chromosomes, or opposite of that you can have fusion where you have 2 chromosomes and they come together and they form a new chromosome. And then you have what are known as insertions and translocations. So, in insertion, a full segment of one chromosome cuts out and gets inserted in a different chromosome at a different position. And in translocation what happens is the same thing, but now you have reciprocal insertion. In other words, you have 2 chromosomes, a portion cuts from here, a portion cuts from here, then that portion is exchanged, which is what I am showing you over here in this figure. So, these are the 7 major types of changes that can happen at the chromosomal level. We have already seen what happens when you have mutation at the level of the entire genome, polyploidy, the condition in which a normally diploid cell or organism acquires 1 or more additional set of chromosomes. Set means the entire set. So, for example, let us say you have a haploid which is N, here we are talking about 3 chromosomes, and let us say then it has doubles, that is diploid condition, you have 3 plus 3 6 chromosome where each chromosome is in 2 copies, and now let us say the ploidy level goes up by even more, by 1 more, then every chromosome is in 3 copies, that is triploid, or 4 copies, that is tetraploid. So, these 2 below, these are the ones that are the polyploids, the 3n and the 4n ones. We have already seen examples of this. Remember, we were talking about the speciation in Primula cuensis. This is exactly the process generating a full 2n equal to became 4n through a process of polyploidy. That is what led to the generation of that new species. We know that there exists a lot of variation in terms of how many chromosomes different species possess. So, an automatic question that comes to one's mind is that if some species has more chromosomes, then does that indicate that it is a more complex species? To understand, or rather to investigate this question, let us look at 4 different species and look at their chromosome numbers. So, here we have 4 species, the jackjumper ant, the human, fern and some kind of a ciliate, it is a one-celled organism. As you can see, the ciliate is a one-celled organism, therefore, it is obviously very simple. This fern is a plant, the jackjumper ant obviously has more kinds of tissue types etc. than the fern, and in terms of complexity one can argue that the human is the most complex. Yet, if you look at the number of chromosomes that they have, here we are showing you the haploid numbers, the n. The jackjumper ant has only 1 chromosome, the human has 23 chromosomes, the fern has 630 chromosomes, whereas the ciliate, which being a one-cell organism is probably the simplest of the lot, that has 16,000 chromosomes. Therefore, this gives us a very important message that the chromosome number does not really predict how complex an organism is. So, after this we are going to look at how mutations impact evolution. So, we are going to study certain properties of mutation which are interesting in this context, and that is what will happen in the next discussion. See you.